Hello, everyone. So welcome to the Protein and Co-Product Graduate Student Oral Competition. This competition was set up by the division actually to give graduate students the opportunity to present and demonstrate the excellence of their research as well as their ability in scientific presentations. So we thank all our volunteer judges and the audience for attending and supporting this competition. So for today, we have got 4 finalists who will present for up to 20 minutes each one. After each student presents, we have about 10 minutes question and feedback session to ask those questions. So if you have any question, please post them in the chat area as well as your comments or congratulatory notes. After all presentation have ended, we will pause the stream for a few minutes to allow for final judges deliberation, and then we will announce the first and second place winners of the competition. So without further ado, let's get started. Our first presenter today is Raliat Abioye. She is a master's student in chemistry and biomolecular sciences at the University of Ottawa in Canada. She obtained her bachelor's degree with honors in biochemistry and molecular biology at the University of Northern British Columbia in 2019, where Raliat was appointed as a research ambassador advocating for research involvement amongst undergraduate students. That's great. Her current research focuses on islet amyloid polypeptide, a hormonal peptide that functions in postprandial glucose metabolism. Her focus is on the identification of food-derived bioactive peptides that have the ability to discourage IAPP, which is the amyloid polypeptide, aggregation through facilitating binding interaction. So the title of her presentation today is Disaggregation of Islet Amyloid Polypeptide Fibrils as a Potential Anti-Fibrillation Mechanism of Tetrapeptide Tngq. So the screen is yours, Raliat, and good luck.
Testing audio.
Oh, sorry. Can you hear me? And welcome to my presentation titled Disaggregation of Islet Amyloid Polypeptide Fibrils as a Potential Anti-Fibrillation Mechanism of Tetrapeptide Tngq. My name is Raliat Abioye, and I'm a student in the Department of Chemistry and Biomolecular Sciences at the University of Ottawa. So just some information about islet amyloid polypeptide, or IAPP. IAPP is a hormonal peptide, and it's co-produced and co-secreted alongside insulin within the beta cells in the islets of Langerhans within the pancreas. It is an intrinsically disordered protein, so it has multiple binding sites, which allows for multiple functions. However, its primary role is in postprandial glucose metabolism regulation. But because IAPP is an intrinsically disordered protein, in the event of uncontrolled expression, this could be highly problematic because in the event of uncontrolled expression, likely as a result of beta cell stress, this can hinder the production and processing of islet amyloid polypeptide. This is problematic because IAPP has an amyloidogenic region, so it's extremely hydrophobic, and this allows for the self-association and subsequent formation of fibrils as a result of improper processing. These fibrils are actually very cytotoxic to beta cells, which is it's highly prevalent in type 2 diabetic patients. So just some background about the Ipp fibrillation kinetics. Fibril formation follows an initial lag phase, followed by an exponential phase, and then a stationary phase. Within the lag phase, we have IAPP monomers, which are either unfolded or misfolded as a result of improper formation after translation. And this exposes the amyloidogenic regions to allow for self-association and the formation of dimers. These formation of dimers indicate the transition into exponential phase, where it undergoes rapid elongation with the self-association of dimers with itself to form oligomers and then protofibrils. These protofibrils can then elongate into mature fibrils, and the formation of mature fibrils indicate the transition into the stationary phase, where the fibrils maintain equilibrium between itself and monomeric IAPP through the slower elongation of the growing ends through association of IAPP. Seeded reactions can resulting in the fragmentation of these mature fibrils can speed up the lag phase to make it that it's essentially not present, and then essentially bypass the lag phase and then continue into exponential and stationary phase. But previous studies have identified the mature fibrils as well as some of the soluble exponential phase species as to be very beta cell cytotoxic. This is facilitated through its ability to permeate through the beta cell membrane and reduce its integrity. As a result, our objectives is to identify a natural anti-IAPP fibrillation peptide inhibitor. Because IAPP has important endogenous roles, we want to identify peptides that can essentially bind IAPP within its earlier phases and prevent the aggregation while also retaining its endogenous functions. Additionally, we would like to elucidate the mechanism of inhibition and its effects on fibrillation kinetics. So our methodology starts off with IAPP pretreatment. Essentially, IAPP, because it's highly amyloidogenic, or it forms fibrils very easily, want to ensure that our IAPP is in monomeric form before we proceed with our characterization of molecular interactions. Characterization of molecular interactions starts off with THT or thioflavin T kinetics assay, where we observe the progression of IAPP fibrillation. Dynamics light scattering allows us to get a better understanding of the types of species and sizes of species present. Circular dichroism allows us to observe the effects of peptide binding to IAPP on its secondary structure. And then molecular docking gives us a better idea of how exactly these peptides are binding to IAPP. Visualization of these peptide fibrils are done with THT fluorescence microscopy to give us just a basic overview of the fibrillation that's happening, but a more focus with transition electron microscopy, which allows us to really see the morphology of IAPP. So into our results, we screened 3 tetrapeptides, YMSV, MANT, and Tngq for its anti-fibrillation activities. So THT, or thioflavin T, is a dye that binds to beta sheet-rich regions, which are highly prevalent within fibrils. And so an increase in fluorescence intensity indicates a progression of fibrillation. So immediately, compared to the control, we see that YMSV and MANT, as well as Tngq, is able to reduce the maximum fluorescence intensity, which indicates that there is a decrease in overall fibrils present. However, MANT and YMSV are to a lesser extent, probably around like 15% of a decrease compared to Tngq, which had a 30% decrease in maximum fluorescence intensity. Additionally, Tngq also was the peptide that strongly decreased the half-time elongation phase, so the time spent within the elongation phase, and also increased the kinetic elongation constant the most, which is why we decided to focus more on Tngq for further downstream characterization. So again, focusing on Tngq, much like the control, we see that there is an increase in fluorescence intensity over time, however, to a lesser extent compared to the control. And then it forms a plateau. However, after about 30 hours, we start to see a decrease in fluorescence intensity. And this could potentially be as a result of disaggregation of the formed fibrils, which we will be exploring later on. But fluorescence imaging of IAPP at 48 hours results into these 2 images, where the control we see longer structures, whereas in Tngq we see much shorter structures, which is indicating that there is some sort of inhibition that is occurring, and also correlating with the decrease in fluorescence intensity over time. So secondary structure characterization with circular dichroism. With IAPP, the control with no inhibitor, we notice a strong double negative peak at 210 and 220 nanometers, which is strongly indicative of alpha helical content. But these negative peaks decrease noticeably after 48 hours. Indicating that there's less of alpha helical content, probably random coils or beta sheet structures that are dominating after 48 hours, and also indicating fibril formation. On the other hand, Tngq in the presence of IAPP at the initial time point immediately decreases the alpha helical content, which indicates that there's potentially a Tngq IAPP complex that is being formed. And even after 48 hours, this the relative secondary structure content remains the same, suggesting that this potential complex also has a stabilization effect on IAPP. So to better understand this IAPP-Tngq complex, we then proceeded with molecular docking. And molecular, just some information on the general domains of IAPP. We first have the membrane binding domain, which allows for IAPP to interact with the beta cell membrane. We have the amyloidogenic region that we discussed previously, and we also have the self-association region, which has been shown to play a very large role in the initial dimerization of monomeric IPP. Previous research has indicated the importance of the amyloidogenic region and actually targeting this in fibril formation inhibition. However, our Tngq peptide inhibitor actually does not associate within the amyloidogenic region at all and prefers a surface binding primarily through hydrophilic interactions as well as hydrogen bonding within the membrane binding domain as well as the self-association region. Its docking score is quite low, and to illustrate how it compares to the other two peptides we selected, MANT and YMSV. YMSV actually had the highest docking score. But focusing on MANT, this peptide preferably has hydrophilic interactions within the protein core surface. And while on the other hand, YMSV, which had the highest score and happened to be the most hydrophobic, bound directly within the protein core, mainly through hydrophobic interactions and some electrostatic interactions. But if you remember, both MANT and YMSV had very moderate inhibitory activity compared to Tngq, which provides a new perspective in terms of inhibitors and where the important region is to bind, drawing more to potentially the membrane binding domain and the self-association region over the amyloidogenic region. So dynamic light scattering results show us the particle sizes, average particle sizes after 48 hours of incubation. And we noticed that there is a threefold decrease in average particle size diameter of IAPP and complexed with Tngq compared to the control. But on the other hand, the average polydispersity index remained the same, which indicates a very heterogeneous environment, largely as a result of various species being formed, either as a result of fibrillation or fragmentation or the Tngq IAPP complexation that we have already confirmed. So TEM, or transmission electron microscopy of the IPP fibrils after 48 hours. Within the control, as expected, we see these well-formed fibrils, which are quite dense and very well branched. However, in the presence of Tngq, we notice, number one, that there's not any branching. They're not as dense, and the fibrils are much more shorter or truncated. Quantification of these fibrils show that in the presence of Tngq, there is an extremely significant decrease in fibril length as well as fibril diameter. As a result, we were able to confirm the inhibitory activity of Tngq. But as mentioned before, we also noticed with the THT kinetics assay a decrease in fluorescence intensity. So moving forward, we wanted to now look into the potential disaggregatory effects of Tngq on IAPP fibrils. So to investigate this, we preformed the IAPP fibrils for 48 hours, and then we added the Tngq, and then observed the progression of fibrillation for another 48 hours. So just looking at the control, at 48 hours, we were able to confirm the formation of extremely dense IAPP fibrillar networks. After 71.5 hours or 23 hours post addition of, in the control, we just added buffer. The networks they progress to be a little bit more dense, but we know that the fibrillation is not as rapid as it is within the elongation phase. And then the same thing remains consistent after 96.5 hours. So this confirms that the equilibrium between the mature fibril and the IAPP monomer has been formed. But on the other hand, in the presence of Tngq, again, we have the formation of these quite dense networks. And we're also able to, after 23.5 hours of adding the Tngq, or 71.5 hours in total, we notice that there are fibrils still present, but interestingly, we also see these amorphous aggregates that are also surrounding the fibrils as well. And previous literature has actually identified these structures to be of exponential phase species. So those are your oligomers or your protofibrils, or potentially even dimers. And then after 48 hours again, or after 48 hours of addition of Tngq, or 96.5 hours in total, we see the reformation of these densely packed fibrils. So just to recap, we have already confirmed the complexation of IIPP with Tngq. But as we mentioned before, we have an equilibrium. So it's likely possible that the complexation of Tngq with IIPP is reducing the overall free IIPP monomers that are present within the surroundings with the fibrils. This could potentially shift the equilibrium of the mature fibrils towards favoring the monomeric IIPP, so that we can reestablish this equilibrium, which is potentially coming off as the disaggregation of these mature fibrils. So in conclusion, we were able to identify that Tngq is a strong inhibitor of IIPP fibrillation, as we see with the 33% decrease in maximum fluorescence intensity, as well as the formation of truncated fibrils after 48 hours. But we're also able to see that Tngq actually also exhibits disaggregatory activities on the preformed fibrils of IIPP, indicated by the decrease in fluorescence intensity, but also these amorphous aggregates that are present after 23.5 hours of addition on preformed fibrils. Finally, we were also able to provide some perspective on the importance of the membrane binding domain, as well as the self-association region of IIPP in inhibiting fibrillation. As previously mentioned, the amyloidogenic region is normally the focused area in terms of targeting IIPP fibrillation inhibition, because it is very instrumental in the fibrillation process. And as a result, especially with the self-association region, which is very instrumental in the initial dimerization of IIPP with itself, it's not as focused. But in our research, we're able to see how Tngq binds to that self-association region and potentially discourages the formation of, or the dimerization, and also results in the decrease in fluorescence intensity. But we're also able to see how the interaction of Tngq with the monomeric IIPP forming complex is able to shift the equilibrium between formed fibrils and monomeric IIPP to favor the production of monomers, which shows up as the disaggregation of IIPP. So I just wanted to acknowledge NSERT and the University of Ottawa for supporting this research, as well as my supervisor and my lab colleagues. And thank you very much for your attention.
Thank you, Raliat, for this great presentation. I think the judges, is the time for your feedback and question, if any. And for those in our audience, please add your question and comment in the live stream chat.
Yeah, I have a question. Okay, thanks, Raliat. Very, very interesting presentation, and really the work, shows that really we can, sort of approach these types of metabolic disorders using very simple tools as peptides, okay, which is very, very promising. My only question is that the TNQ, TN, yeah, anyway, your peptide is highly hydrophilic. Do you think really it can be absorbed very well in terms of practicality? So if somebody actually wants to use this, can the peptide really be absorbed? Have you done any work in terms of bioavailability of that peptide, because it's highly hydrophilic?
Yes, that is true. We have done in silico characterizations, which confirm that it is very low. It has a very low bioavailability score. But however, Tngq is food derived. It's actually from rice bran. So it is possible that the through digestion, it can be released within the stomach. However, it is also important, or a great future research, to also look into potential delivery methods, such as encapsulation, to allow for a more targeted delivery to the stomach.
Okay, thank you.
So any other question?
I do. Thank you, Raliat, for your presentation. My question has to do with the your finding that the TNGQ maintained the alpha helical conformation of the monomeric IAPP. What is the significance of that particular finding?
Yes, so maintaining the alpha helical conformation of IPP is very important, because it's said to be the state at which it's most stable. So when it's not in the alpha helical content, or in the structure, it favors to be essentially a random coil. And this configuration can allow it to not only expose the amyloidogenic region and the self-associating domain, to allow for the self-association and progression of fibrillation, but because it is an intrinsically disordered protein and it has multiple binding sites, the random coil configuration can allow it to have off-targeted binding when it's not necessary. So having a peptide or any type of protein or any type of molecule that can bind to and maintain that alpha helical structure, it kind of forms or acts as a stabilization effect, or a chaperone, if you will, to prevent its association with other things that are not necessary at that time.
Thank you.
Thank you. I think, Hao-Chen, you have, yes, please go ahead.
I have a question. Very, very interesting work and very impressive. So I have two questions. My first one is I think you pretty much mentioned about the mechanism. I noticed you use a term called disaggregation. So by using this peptide, is the mechanism all more about degrading the fibers, or more about preventing the formation of the fibers?
Actually, if possible, both would be very great, especially because within type 2 diabetic patients, these amyloidogenic deposits are quite prevalent. So if we have an agent that can disaggregate these fibrils, then this could prevent the ongoing beta cell cytotoxicity that is present. But at the same time, as a preventative measure, if we could have an agent that binds the monomeric IAPP kind of as a chaperone to also discourage self-aggregation, both mechanisms are equally important.
Sure. Okay. So particularly for your work, the materials you use are very much about preventing the formation of aggregation, right? And because of the binding of the what's the name Tngq, right? So I also noticed that the difference between the Tngq treated and the control. So regarding the curve, right? So are they? So I don't see any deviations there. So are they statistically significant for the two separated curves between control and the treated sample?
So you're asking if I guess the maximum fluorescence intensity decrease is statistically significant?
Yeah.
Okay. I didn't actually calculate statistical significance. In general, we perform all of our assays in triplicates. However, when it comes to actual modeling, the software actually averages all of the runs and then does it. So because we're not able to do the standard deviation, it's very difficult to also calculate the statistical significance.
Yeah. Okay. Yeah. Thank you.
Okay, thank you. I don't see questions in the chat. I just have maybe one last question for you, Raliat. Is just what is actually the physiological impact of these fibrils accumulating in diabetic people? I mean, what will be the impact on their health?
Yes, so with the formation of these fibrils, it's resulting in the beta cell cytotoxicity. So it's killing beta cells. But if you remember, IAPP and insulin, they're co-produced and co-secreted at the same time. So when we have a decrease in beta cell mass as a result of cytotoxicity, the remaining beta cells have to essentially overwork themselves to compensate for the decrease in beta cell mass as a result to increase insulin production. But since insulin is also being produced, IAPP is also being produced at the same time, which is increasing the overall presence of IAPP within the beta cells. And it's kind of like a cycle because we're producing IAPP and that's those fibrils are killing the beta cells, and then the beta cells are reproducing IAPP, and then so on and so forth. So the physiological importance would be to essentially chaperone the IAPP so that it's not able to form these fibrils, which would give the cells the ability to actually degrade them before they form fibrils, and then also reduce the beta cell cytotoxicity and hopefully increase beta cell mass and insulin production and glucose regulation in the long run.
Okay, great. Thank you very much. Okay, I think that brings this one to the end, and it's time for our next presentation. Thank you.
Thank you.
Okay, so our second finalist today is Yifu Chu, who is a PhD candidate in Dr. Lingyun Chen lab in the Department of Agricultural, Food and Nutritional Science of the University of Alberta in Canada. His research is mainly focused on the generation of functional microgels based on biopolymer materials and exploring their applications in food and biomaterial fields. So Yifu will present today on the effect of uniform whey protein microgels on oil in water emulsion property improvements and their potential application as fat replacers to prepare fat-reduced food products. So welcome, Yifu, and good luck.
Thank you. Thank you, Dr. Hosing. Hi, everyone. My name is Yifu Chu, and I'm a PhD candidate in Dr. Lingyun Chen's lab at the University of Alberta in Canada. And today I'll share with you part of my PhD project, which is mainly about the effect of uniform protein microgels on oil and water emulsion property improvements and their potential application as fat replacers. So according to WHO, obesity has tripled over the past 40 years. And the contribution of the calorie-dense fat to this global crisis cannot be ignored. So design fat replacer to mimic the function and the sensory properties of fat can be a potential strategy to decrease calorie intake. So as you can see in the middle figure here, the fat presents in either milk, yogurt, or cheese in the form of small fat droplets. So fat droplets are highly related to food sensory qualities, and they provide smell, flavor, and most importantly, they provide texture like viscosity, and can work as oral lubricants to generate creamy mouthfeels. So today, the application of fat replacers in industry is mainly focused on improving the viscosity by adding thickening agent like polysaccharides. But there is a limited investigation on replicating the creaminess brought by fat droplets, and this concept has not been really applied in real food model nor industry so far. So protein provides much less calorie than fat. So the hypothesis is the protein-based microgels can be prepared with similar size and shape as fat droplets, as you can see in these two figures, which have the potential to be applied as fat replacers to replicate the functionalities of fat droplets in food products. The next I'm going to talk about what is microgel. So we all know that the gel is basically on the microscopy level with connected networks of a kind of polymer to hold large amount of solvent in the structure. For example, protein-based gels, just as shown in this figure. Then let's imagine if we can express this gel-like feature on the microscopy level, say from several micrometers down to several nanometers, then we'll get a special type of soft colloidal system, which have both gel-like and particle-like features. So as we can see in the cross-section SEM images here, essentially microgels and macrogels have similar internal structures. So there are basically three methods to prepare protein microgels, like protein macrogel shell breakdown or protein self-assembly by heat treatment under certain pH, or the microemulsion template method. But protein microgels prepared by these current methods have some limitations for commercial use. For example, it is still challenging to make microgels with narrow size distribution. The size greater than around 20 micrometers are perceived as grainy and may increase friction in mouth. And high energy input is normally being used to control the size distribution of the obtained protein microgels, like high pressure homogenizer. And the irregular shape of the current protein microgels, normally from the protein macrogel shell breakdown method, may also increase friction. So here I would introduce a new method to prepare protein microgels from water emulsion droplet, which is from protein polysaccharide phase separations. So whey protein will be used in this study, and its main component is beta-lactoglobulin with molecular weight of around 18K Dalton. And the whey protein has excellent gelling property when heating over 75 degrees. The exposed cell groups from cysteine can form disulfur bond, which contribute to the gel networks. Then we'll talk about how to form a protein polysaccharide water emulsion. So as you can see in this figure, there are essentially two different types of the protein polysaccharide interaction. So first, when the two biopolymers are oppositely charged, the protein and the polysaccharide were associated with each other through electrostatic interaction to form a one-phase system. And when the two biopolymers carry similar charges, then the protein and polysaccharide will repel with each other to form a two-phase system or segregative phase separation, which will then trigger the formation of water-in-water emulsions. So one would expect that the gelation of the water-in-water emulsion droplets can be used to prepare spherical and uniform protein microgels without using any surfactant and high energy input because of the ultra-low interfacial tension of the water-water interface. So the overall hypothesis is that protein microgels can be used as fat replacers to mimic fat droplets in food products. And to be specific, the water-in-water emulsion can be used to prepare uniform and spherical whey protein microgels. And the protein microgel addition can enhance the triple rheological properties of the fat-reduced emulsions. And this protein microgel system can be applied as fat replacers to develop fat-reduced foods. So today we're going to have three objectives. The first is to develop uniform and size controllable whey protein microgels by heat gelation of water-in-water emulsion droplets from protein and polysaccharide segregative phase separation, and to study the contribution of microgels at the interphase and bulk phase to the improved emulsion stability and texture, and to explore their potentials to mimic the functionality of oil droplets in the emulsion systems. So first, I will talk about the first objective, about preparing uniform and size controllable whey protein microgels. And in this study, uniform whey protein microgels were formed by heating the mixtures of the whey protein solution and alginate solution at 90 degrees for 10 minutes at pH 7 when both biopolymers were negatively charged. And the microgel size can be controlled by adjusting the whey protein and alginate solution volume ratios. The microgel size and microstructure were tested, and the properties of emulsion stabilized by the microgel system were also tested. The next, I will show some representative figures for this objective. So as you can see in this phase diagram, with x-axis showing the alginate concentration and protein alginate volume ratio in y-axis. The figures show that the alginate concentration had to reach a critical value to form whey protein microgels. At alginate concentration of 2%, by changing the protein and alginate volume ratio from 2:1 to 1:4, the microgel size decreased gradually, as you can see in the right side of this figure. And based on the size measurement, I found that the microgel size can be precisely controlled to be 1 micrometer to 3 micrometer, and then to 6 micrometers by adjusting the protein and alginate ratios with narrow size distribution. And the ICM image also showed a homogeneous microgel size and a spherical shape, and the cross-section image of an individual microgel showed a cross-linked gel networks. And the figure below showed that the emulsion stabilized by the microgel system had longer stability and smaller droplet size and higher viscosity when compared to the emulsion stabilized by the whey protein and the polysaccharide, but without microgels. So in summary, uniform and spherical whey protein microgels can be prepared by the water-in-water emulsion method without high energy input to control size distribution. So when we talk about the gap, we're trying to find a method to control the shape and size distribution, but without high energy input. And now we've achieved that. The microgel size can be controlled by adjusting protein and alginate ratios, and an emulsion stabilized by the microgel system showed longer stability and a higher viscosity. So here comes one question that what is the underlying emulsion stabilization mechanisms of the whey protein microgel system? So before we study the rheological or lubrication property of these microgels, we'd better understand the behavior of these microgels in the emulsification, like whether it is adsorbed at the interface or dispersed in the bulk phase. So here comes objective 2 to study the contribution of microgels at the interface and bulk phase to improve the emulsion properties. So based on the experiment, the microgel system in objective 1 is actually a hybrid system of microgels, unconverted protein molecules, and polysaccharides, and the microgel conversion rate is around 50%. So to understand their individual contribution to the emulsion stabilization, this hybrid system was separated and then recombined to different microgel system formulations, including isolated microgel alone, and a microgel combined with polysaccharide, microgel combined with protein molecules, or a system containing all three components, which is the same as the original system. And the individual properties of these formulations and oil in water emulsion stabilized by these formulations were tested. So next, I will show some representative data for objective 2. So the confocal images here show the emulsion microstructure with microgels in red color. So as you can see, the emulsion stabilization mechanisms of these formulations can be basically divided into two scenarios. So in scenario 1, when there is no protein molecules in the formulations, it is clear that the microgel are dissolved at the interface to form a kind of Pickering emulsion, as you can see in these two figures. And in scenario 2, well, the protein molecules are in the formulations, it can be seen that the interface is covered by a thin red film, which is the protein molecules, where the microgels are in the continuous phase, which means the microgels no longer work as emulsifier when protein molecules are in the system. And this can be explained by the individual tension data right here, that the protein molecules can much more fastly adsorb to the interface to decrease individual tension than microgels can do. And that is because of the nano size and amphiphilic nature of protein molecules compared to the micro-sized microgels. So today we're going to focus more on the scenario 2 when the microgels are in the bulk phase, because in our objectives, we prefer the microgels can work in the continuous phase to replicate the functionalities of the fat droplets. So here is the next objective to study the effect of protein microgels on improving the emulsion texture properties. So first, I will introduce two functionalities of fat droplets to generate fat-related attributes, which should be highlighted. So first is the thickening property, where fat droplets act as fillers to enhance the rheological properties to provide thickness perception in mouse. And in this case, the gap between tongue and palate is normally larger than 100 micrometers, then the rheological properties will be dominated in this case. And the second is the rubrication property, where fat droplets act as oral lubricants to decrease the friction between tongue and palate to provide creaminess and smoothness perception. And in this case, the tongue and palate contact with each other. So the tribological property will be focused on to study the two interacting surfaces in relative motion. So in this objective, the protein microgels will be used as a kind of fat replacer in emulsion system, and the emulsion textures will be focused on. So in this objective, the rheometer will be used to measure the tribological properties of the emulsion system with or without microgels, which can be a promising strategy to predict the perception of food. So viscosity is mainly related to the thickness perception of food, where the physical elasticity is related to the stiffness, and the tribological test can be used to predict the creaminess and the smoothness perception of food. And these two figures show that the increased microgel content in the continuous phase can enhance the rheological properties of the fat-reduced emulsion. And the emulsion with 30% oil with 1.5% of microgels show the comparable elasticity and even higher viscosity to that of emulsion with 50% oil but without microgels. So the confocal image here also showed that the microgels can work as a filter in the emulsion system that can improve the rheological properties, and it can also help prevent oil droplets flocculation to increase emulsion stability. So in our lab, we're now working on testing the tripological properties of the protein microgels and the emulsion with microgels. And our hypothesis is that the protein microgels with spherical shape and the uniform size distribution can work as an efficient or a lubricant to mimic the lubrication properties of the fat droplets. So in conclusion, increased microgel content in the bulk phase can make fat-reduced emulsion have comparable rheological properties to emulsion with higher fat content, so which can be beneficial to the fat-reduced food development. So our long-term objective is trying to apply these spherical and uniform protein microgels as fat replacers into real food models, especially emulsion-based food products. So one example here is yogurt, which is a kind of dairy semi-solid. Full-fat yogurt can have a fat content over 8%, and reducing fat may cause quality issues like low viscosity, firm texture, and low stability, which can be generally addressed by adding thickeners like polysaccharide and protein. And most importantly, reducing fat may also cause a reduction in creaminess and smoothness, which is directly related to the customer acceptance. So protein microgel will be expected to replicate these fat-related attributes for the fat-reduced yogurt products in the future. So we are also trying to explore other potential emulsion-based food models like liquid or semi-solid food, including salad dressing or yogurt, and the emulsion gel or solid food, such as ham and cheese. And we expect that the microgels can benefit the healthy food industry development to help people decrease the total fat intake. So in conclusion, the uniform and size controllable protein microgels were prepared by heat gelation of boiling water emulsion droplets. And the microgels can be surface active to form pickle emulsion or surface inactive to disperse in the continuous phase, depending on whether there's any protein molecules in the system. And increased microgel content in the bulk phase can make fat-reduced emulsion have comparable rheological properties to the emulsion with higher fat content. So this project leads to a novel method to prepare uniform and size controllable protein microgels without high-energy input to control the size distribution. And it will also help understand the interfacial properties and the adsorption behaviors of the microgel system. And it will also help generate knowledge of evaluating the textural properties of emulsion containing microgels. And this study can benefit the fat-reduced food development to provide fat-reduced food options for healthy outcomes, such as reducing calorie intake to help control the world obesity epidemic. I really appreciate the support from my supervisor, Dr. Lingyun Chen, and all my lab mates, and I also thank all the audience to join together with me in this presentation. Thanks.
Thank you, Yifu, for this nice presentation. So please, the judges, it's time for your feedback and questions.
I'd like to go first.
Yes.
Thank you, Yifu, for your presentation. I have two questions for you. The first one is, what is your experimental and analytical replication?
Uh, um, I, 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 I think I don't really get you. What do you mean replication?
Essentially, how many times did you do your to get your result? Yes,
like a replication.
For each phase, did you do it 2 times, or did you work with 2 batches? For analytical replication, did you do triplicates, duplicates for the test that you did?
Yeah, for most of the results, I do triplicate.
Okay.
Yeah.
My second question, is there a particular type of protein or protein structure that would work best in the microgel system that you developed?
So far, in our lab, I just tried whey protein. So, and we are also looking like any chance to transfer to the plant protein, because plant protein is always like more attractive than the animal protein in recent years. I think the plant protein also gonna work as long as they can, have electrostatic repulsion with the polysaccharide, and you need to get a critical concentration for both biopolymers to form the gel structure. I think, yes, theoretically, all protein is going to work.
Thank you.
Yeah, thank you.
Hello, may I go second?
Sure.
Hi, Yifu, this is impressive work. Thank you for your presentation.
Thank you.
I have a few questions. The first one is about your emulsion stability. I think this is can't remember if this was your phase 1 or phase 2 study. So how did you characterize the emulsion stability, and how do you get the conclusion, say, your Pickering emulsion was more stable than the control?
There's some misunderstanding. I'm sorry. Like in the phase 1, like the emulsion is actually not a Pickering emulsion. It's a molecular stabilized emulsion. It's stabilized by the protein molecules rather than the microgels. So in terms of the stability, how I characterize is, like, firstly, to check whether it has phase separation during the storage. And the second is to check their oil droplet size to see whether there's a significant change of the oil droplet size. If there is a significant change of oil droplets, which means there's coalescence of the oil droplets happens, which means it's not stable. So in our experiment, the stable emulsion, firstly, we're not show any phase separation during the storage, and the droplet size will not change significantly during the storage as well.
So what's the mechanism that gave you this stability? Because I couldn't remember the particle size of your oil droplets, but I think most of the particle stabilized emulsion normally give you relatively higher particle size, right? Due to Stokes' law, you should have some creaming over the time, but for your system, it's quite stable. What the mechanism behind that?
Well, in terms of the mechanism for why the emulsion containing microgels is stabilized by the microgel system is stable, is because the microgels in the bulk phase can work as a fillers that can firstly increase the viscosity of the bulk phase, which can prevent slow down the rate of the oil droplets to flocculation or coalescence. And the microgels in the continuous phase can also work as like a hindrance to stop the flocculation of the oil droplets. That's also can help increase the stability of the emulsion system. So,
but isn't the microgel particles are colloidal particles? They supposed to somehow sediment over the time? I can understand they may increase your viscosity because of the increase the solid content, but over the time, right? They not supposed to sediment. Or they could
during the
system.
During the storage, precipitation. Yeah. I didn't find any precipitation during the storage time.
What's the reason? What's the reason for that?
First, I think so I think the viscosity, the increase of viscosity can explain largely explain why the system is stable but without creamy or precipitation.
Is that also partially due to the alginate you added?
For sure. I forget about that. Yes. Alginate also is like a thickener, like it can increase the bulk viscosity. But we have a control group that show the emulsion is stabilized by the whey protein and the alginate, but without microgels, they are not very stable. So microgels may also play a very important role in terms of the emulsion stability.
Okay.
So they have a kind of like a synergistic effect between the alginate and the microgels.
Yeah. Okay, thank you. My last question is: so you didn't show the tribology results. Are you doing that or?
Yeah, we are doing it in our lab. We don't have data to share. So when we get the data, we can have more comments.
Yeah, it will be very interesting to see the tribology results, to see the smoothness of the system.
Yes.
Okay, thank you.
Thank you. Thank you for your question.
Okay, thank you very much. I just took questions.
Yeah.
Very good work, very interesting. I like this type of very mechanistic work, looking at the fundamental basis of how proteins work. My question is, what is a water in water emulsion?
Oh, the water in water emulsion is, which means like is from the protein and the polysaccharide phase separation mechanism. It's like a protein solution dispersed in the alginate continuous phase.
So you have the protein that is separated, right?
Yes. So right.
That cannot be an emulsion. An emulsion is a continuous phase. So if you have certain parts that is separated, then that cannot be an emulsion, because I've never heard about a water emulsion. An emulsion is the dispersion of an insoluble phase in terms of droplets, okay, in another phase that then forms a continuous phase. So I'm just curious how you define a water in a water emulsion.
Yeah, so water in water emulsion in our study is definitely like the protein solution droplets dispersed in the alginate continuous phase. But this solution, this water in water emulsion is not stable. Of course, it will form phase separation maybe after overnight. It will phase separation, but what we do to make microgels based on water in water emulsion is to when it forms water in water emulsion, we use the protein will form microgel, like it will form microgels during heat treatment. So what we do is to make them gelled before they form phase separation.
Yeah, okay, well that's fine. Maybe we'll discuss that later on, but I don't agree with you that there's something called a water in a water emulsion. Okay. But anyway, the second question very quickly, how does your work differ from the fact that we already have a commercial microparticulated protein as a fat replacer. It's called Simplis. It's also a microparticulated protein, right, from milk and egg that is being used as a fat replacer. I'm just curious how, what is the novelty here? How is your work different from what already exists as a commercial microparticulated protein?
Yeah, thanks for that question. Like, I've seen a lot of research papers demonstrate that the microparticular whey protein is that right? It can be worked as an efficient lubricant that can have a creamy mouthfeel in some food products. So the difference of our microgel between the microparticular whey protein is that our microgel is kind of soft colloidal system. So, and our microgel is very, the size is very, it has a narrow size distribution. So I think it's gonna have, be more efficient in terms of the decrease the friction coefficient, and the solid particles. For solid particles, if the size is not homogeneous, it may increase the friction. But in terms of the soft colloid, like microgels, it can decrease that effect. I think the microgel is going to be more efficient in terms of decreasing the friction. But we still need more experiments to testify. Just microgel, I think, has a lot of potentials in terms of the fat replacers application.
Okay,
thank you. Thank you.
Yeah.
Thank you. I think our time is up for this presentation. Thank you, Yifu.
Thank you.
Thank you.
Thank you for the question.